Welcome to our latest deep dive. Whether you're prepping for a high-level neurobiology meeting, trying to catch up on the latest in cognitive science, or you're just insanely curious about the hidden machinations of your own mind, you are exactly where you need to be.
You really are. And today we are turning our attention to the back of your skull.
Right, the cerebellum.
Yeah.
We're diving into a fascinating 2022 paper published in Nature Neuroscience by Liu and colleagues. And it really fundamentally changes how we view the human cerebellum.
It's a piece of research that truly shifts the paradigm. Because for the longest time, the scientific consensus regarding the cerebellum, which literally translates to the little brain, by the way, was that it acted as a relatively simple motor control and error correction center.
Right, the classic textbook view.
Exactly. The idea was that if you stumbled while walking or, missed a tennis backhand, your cerebellum was the thing adjusting your coordination in real time. But this paper completely dismantles that limited perspective.
It really does, because science has recently realized that this little brain actually plays a massive role in complex cognition, and it's even deeply implicated in developmental conditions like autism.
Which is huge.
But the mission of our deep dive today is to explore a specific, groundbreaking question the researchers asked. We know that the neocortex, that wrinkly outer layer of your brain, has these beautiful, well-documented structural gradients.
Yeah, it possesses a distinct hierarchy of growth.
Right, and myelination that shifts predictably from basic sensory processing areas all the way up to high-level cognitive areas. So the core question Liu and colleagues posed was, does the cerebellum have this kind of gradient too?
And to get to the bottom of that, the researchers had to combine massive multimodal datasets. I mean, we are talking standard structural MRI, quantitative MRI, functional MRI, and even postmortem proteomics.
A mountain of data.
A literal mountain. And what they proved is that the cerebellum isn't just some structurally uniform blob of tissue at the base of the brain. It's a beautifully complex, multifaceted gradient with a distinct topography that shapes how we develop.
From childhood right into adulthood.
Exactly.
Okay, let's unpack this by looking at the adult cerebellum first. The static map, so to speak. To map out the structure, the researchers used the T1 weighted over T2 weighted MRI ratio.
Or just T1W over T2W.
Right. And assuming you're familiar with standard MRI scans, taking the T1 image and mathematically dividing it by the T2 image is such a clever workaround for visualizing brain tissue.
It's an incredibly elegant mathematical trick. Because with a T1 scan, fatty tissues like myelin show up bright, while a T2 scan tends to highlight fluid.
But both of those individual scans suffer from inherent magnetic biases from the scanner itself.
Yeah, what we call spatial inhomogeneities. You get these background shadows that can skew the data.
So by taking that ratio, you're essentially canceling out the scanner's background noise. Think of it like using two different flashlight filters. You end up with a highly specialized, clean magnifying glass, specifically for myelin.
And myelin, of course, is that fatty, white insulating sheath wrapping around nerve fibers, which allows electrical signals to travel at really high speeds.
The more myelin in a specific area, the higher that T1 over T2 ratio is going to be.
And when they applied this myelin proxy to a massive data set, 1,018 adults from the Human Connectome Project, all between the ages of 22 and 37, the resulting map was staggering.
Staggering how?
Well, the myelin wasn't randomly scattered, nor was it a flat, uniform distribution across the cerebellar folds, which are called lobules. It formed a highly specific, continuous topography.
If you map it out on a graph, it looks exactly like an S-shaped roller coaster.
That is the perfect way to visualize it, an S-shaped roller coaster.
Tracing that roller coaster provides such a clear picture of the anatomy. The ride starts high up in the anterior regions of the cerebellum, specifically lobules I through 3.
Right, the regions heavily involved in immediate primary sensory and motor representations.
Here, the myelin content is extremely high, meaning the neural wiring is heavily insulated for rapid-fire processing. But as you move structurally toward the posterior of the cerebellum,
Into the higher level association and cognitive regions,
the myelin levels take a steep dive.
they drop all the way down to a nadir, the absolute lowest point on the structural roller coaster in a region known as crust 2.
Crust 2.
Yeah. And then, continuing that S shape, the myelin levels slowly rise back up as you move counterclockwise toward lobule X, before finally dropping again at lobule X.
It's a striking S-curve that maps perfectly onto the physical anatomy of the little brain.
It is.
So if I'm visualizing this correctly, let's do a quick subtopic recap and bullet summary here so we can lock this in. First bullet, the T1W over T2W ratio is our powerful proxy for mapping myelin.
Second bullet, the adult cerebellum features a highly distinct S-shaped structural gradient.
And the third bullet, anterior sensory regions have heavy insulation, while the posterior cognitive regions, like Chris II, have much less.
Spot on. Which tells us that the physical structure of the tissue perfectly reflects the complexity of its function.
Heavy myelination is great for fast, rigid, hardwired motor responses.
And less myelination in the cognitive areas likely allows for more synaptic plasticity, the flexibility needed for complex thought and association.
That raises a really fascinating question for you, the listener, to ponder. If you picture your own cerebellum, why do you think the areas processing immediate sensory input need thicker wiring insulation than the areas pondering complex thoughts?
It makes a lot of sense when you think about the evolutionary need to pull your hand away from a hot stove instantly.
Versus the time needed to, say, solve a math problem.
Exactly. That evolutionary perspective is crucial, especially when we look at how this structure forms over time. We have this beautiful S-shaped map in adults, but we certainly aren't born with that roller coaster fully built.
Which brings us to the developmental gradient. To figure out how we actually build that map, the researchers analyzed the Developmental Human Connectome Project dataset.
This included 519 children and adolescents ranging from ages 9 to 19.
And they applied that exact same T1 over T2 myelin metric to watch the brain grow in real time.
The growth dynamics they uncovered show a massive divergence between different parts of the cerebellum. The anterior sensory lobules, the ones with high myelin in adults, showed completely flat or even slightly negative developmental trends in the kids.
What surprised me the most about that growth mechanism was just how stagnant those sensory lobules are in comparison to the rest of the brain.
They really are.
They're essentially structurally mature by the time we hit childhood.
It comes back to basic survival. You need your primary motor skills and sensory processing online very early in life. You cannot afford to have those areas under construction when you're learning to walk and interact with the physical world.
But the posterior higher-level cognitive lobules, like Crus's S and Crus's II, tell a totally different story.
They show aggressive proliferation and tissue growth throughout late childhood and deep into adolescence.
The cognitive regions are essentially playing a massive, prolonged game of catch-up. And the researchers highlighted this with a really interesting statistical detail regarding interlobular heterogeneity.
The standard deviation.
Right, the structural difference between all these varying cerebellar lobules. In younger kids, the cerebellum is highly contrasted, extreme highs in the sensory areas and extreme lows in the cognitive areas.
But this heterogeneity actually decreases linearly.
By about 1.94% per year from childhood to adulthood.
That 1.94% annual decrease is the mathematical signature of the posterior regions catching up to the anterior regions. The cerebellum starts out with extreme structural spikiness.
But as those cognitive association areas aggressively grow and myelinate during your teenage years, the overall landscape smooths out
Into a much more cohesive, integrated network.
It really shifts the perspective on what's happening inside a teenager's head. But it also brings up the classic nature versus nurture debate. How much of this protracted cognitive growth is just the environment?
Like learning new things, going to school.
Exactly. And how much is baked into our DNA?
What's fascinating here is that the researchers address that by looking at twin data within the dataset. By comparing monozygotic identical twins with dizygotic fraternal twins, they calculated a heritability index for these regions.
And they found that the structural tissue values in those late developing posterior cognitive lobules, specifically Crus's II, and VII, are highly heritable.
So there's a strong inherited genetic blueprint guiding the construction of these specific cognitive areas. It's not just about the math homework you do. The timeline of this growth is literally coded in your genes.
Let's do another quick subtopic recap and bullet summary here. First bullet, sensory lobules mature early, but cognitive lobules grow aggressively throughout adolescence.
Second bullet, the structural differences that heterogeneity between lobules shrinks by nearly 2% a year as we age into adulthood.
And third bullet, this posterior cognitive growth is strongly guided by genetics.
Absolutely.
So listener, knowing that your cognitive suburbs are still under heavy construction well into your teens, how does that reframe your view of adolescent learning and behavior?
It certainly makes me a lot more forgiving of my teenage self.
Same here. But, as robust as the standard MRI data is, it's still relying on a proxy. It's somewhat qualitative.
True.
So to validate this map, here is where it gets really interesting. The researchers turned to quantitative MRI, or qMRI, and postmortem proteomics.
This was a vital methodological step. A standard T1 over T2 ratio suggests relative differences in myelin, but quantitative MRI gives you absolute physical units of measurement.
You can compare these metrics directly across different individuals and different scanners.
Almost like taking a precise temperature. They utilized two specific qMRI metrics for this: relaxivity, known as R1, and macromolecular tissue volume, or MTV.
I want to make sure we really grasp the physics behind these metrics, starting with relaxivity, or R1.
Let's do it.
When the MRI magnet excites the protons in your brain tissue, those protons are knocked out of their magnetic alignment. R1 measures the exact rate in hertz at which those protons relax back to field alignment.
And that rate is highly sensitive to the presence of hydrophobic material.
Specifically, the lipids that make up myelin, because water relaxes differently than fat.
By mapping the R1 relaxivity, they found it decreased significantly as you move from the anterior to the posterior lobules.
Which perfectly validated the original T1 over T2 ratio map using an absolute physical metric. The lipid-heavy myelin really is dropping off in those cognitive regions.
Then there's MTV, macromolecular tissue volume. This measures the actual physical fraction of a brain voxel, which is like a 3D pixel of the scan that is made up of physical tissue.
Things like proteins and lipids rather than just water.
Measuring changes in MTV is incredibly difficult, but when the researchers compared qMRI scans of 35 children against 29 adults, the results were definitive.
They proved that cruci, that posterior cognitive region we keep mentioning, physically grows in macromolecular volume over time.
It grows by over 0.1 microliters per voxel.
While the anterior sensory lobules I through 4V, essentially don't change at all in volume.
It is hard, physical proof of localized tissue growth happening during adolescence.
That is a staggering level of precision for a living human brain scan. But they didn't stop at neuroimaging. They took the validation all the way down to the microscopic molecular level.
By analyzing postmortem cerebellar tissue from a 3.5-year-old child using mass spectrometry.
Mass spectrometry allows researchers to identify and quantify the specific proteins enriched in different tissue samples. And when they compared the protein expression from the anterior lobules I through 5 against the posterior cruci at the second,
the molecular differences were stark.
If we think about lobules I through 5 as the heavy machinery factory floors of the brain, the mass spectrometry data backed that up entirely.
These anterior areas were absolutely packed with myelin basic protein, or MBP.
Alongside proteins essential for building synapse structures and neurite growth factors.
It's an environment completely dedicated to building thick cables and securing heavy, permanent connections.
But in contrast, the protein environment in CRUST-10 is entirely different. CRUST-2 acts as the management office.
The proteins enriched there weren't for heavy wiring. They were proteins primarily involved in DNA and RNA regulation.
These are cellular planning and genetic management tools, transcription factors that dictate future growth and plasticity.
Molecular processes that aren't even readily detectable on a bulk MRI scan.
Time for our subtopic 3 bullet summary. First bullet, qMRI metrics like R1 and MTV prove the posterior cerebellum physically grows more tissue over time.
Second bullet, mass spectrometry reveals anterior lobules are rich in myelin proteins.
And the third bullet, posterior lobules are rich in DNA and RNA regulatory proteins.
It's just a stunning convergence of evidence. You have bulk imaging data from living populations perfectly aligning with cellular protein data from a postmortem sample.
Which begs an important question for you listening. If we can trace brain differences all the way down to the individual proteins, what could this mean for treating neurological conditions at the molecular level?
It opens up an entirely new avenue for targeted therapies.
Absolutely. But having mapped the physical hardware and seen the proteins building it, what about the software? Does this structural gradient actually alter the way the brain fires?
That is the ultimate test of the gradient's relevance. To measure the functional software, the researchers utilized resting state functional MRI, or RSF MRI.
This technique looks at the brain's intrinsic activity while a person is just lying in the scanner, not performing any specific cognitive or motor task.
They specifically focused on a metric called FALFF,
which stands for the fractional amplitude of low-frequency fluctuations. Try saying that 5 times fast.
Not a chance.
To understand FALFF, you have to think of the brain's neural signal like a complex symphony with many different frequencies of sound playing at once. FALFF is the ratio of the low-frequency power, specifically the oscillations between 0.01 and zero point 0.08 hertz, compared to the full-range power in the brain's electrical signal.
These low-frequency fluctuations are incredibly important because they are closely tied to the brain's default mode network and intrinsic functional states.
FALFF acts as a highly stable, reliable functional fingerprint of local brain processing. It tells us how active a specific region is when it's just idling.
And when they mapped this functional software across the cerebellum, the topography was a revelation. It formed the exact same S-shaped gradient across the lobules, but perfectly inverted in sign compared to the structural T1W over T2W gradient.
It's a precise mirror image.
In the anterior regions where the structural myelin ratio goes up, the functional low-frequency amplitude goes down.
And in the posterior cognitive regions where myelin is sparse, the FALFF signal is significantly higher.
And this functional software has a remarkably tight developmental coupling to the hardware.
As the brain grows from childhood to adulthood, it's the posterior cognitive lobules that show the most dramatic functional decreases in FALFF.
What is truly remarkable is how synchronized this process is within a single person over time. If you track an individual's structural gradient alongside their functional gradient as they age, the two metrics do not drift apart.
They remain tightly mathematically coupled.
The researchers calculated an R-squared value, a statistical measure of how well two variables correlate, and it remains stably above 0.5 throughout their entire developmental trajectory.
Let's do our final bullet summary here. First bullet, FALFF measures the low-frequency resting activity of the brain.
Second bullet, the functional gradient perfectly mirrors but inverts the structural S-curve.
And third bullet, structural maturation and functional maturation are deeply synchronized throughout childhood.
It provides a unified theory of cerebellar development. The hardware dictates the software, and they mature in lockstep.
Think about your own mental focus for a second. How does it feel knowing that the physical growth of your brain tissue is directly tethered to the electrical rhythm of your thoughts?
It grounds our understanding of psychology and cognition in very tangible, measurable biology.
So what does this all mean? As we bring this deep dive to a close, let's distill the big picture.
The cerebellum is emphatically not just a uniform motor center.
It possesses a prolonged, highly coordinated gradient of structural and functional development from sensory to cognitive areas.
And if we connect this foundational science to the bigger picture, the clinical implications are profound. This multifaceted gradient serves as a crucial new biomarker.
Because we know from vast amounts of clinical research that atypical myelination directly leads to abnormal brain function.
Meaning this S-shaped gradient could be the key to identifying and understanding the mechanisms behind developmental disorders like autism.
It provides a tangible roadmap. Conditions like autism spectrum disorder are increasingly viewed as disorders of developmental timing and neural connectivity.
If the posterior cognitive regions of the cerebellum fail to catch up, or if the functional FALFX signal doesn't couple correctly with the structural growth, clinicians finally have a map of what to look for.
It moves our understanding of these conditions away from purely behavioral observations and grounds them in highly specific, measurable neurodevelopmental trajectories.
It really is a roadmap to the little brain that we desperately needed.
As we wrap up, I want to leave you with one final provocative thought to mull over. We've established that the cognitive association regions of the cerebellum, those posterior lobules like crests of theirs and crests of first, show the most genetic heritability and the most prolonged growth.
Which strongly suggests they are tied to our rapid evolution as humans.
Exactly. So next time you learn a complex new skill or reflect on a deep memory, ask yourself, is my neocortex doing the heavy lifting, or is the secret engine of my little brain pulling the strings in the background?
It's a question that neuroscience will undoubtedly be exploring for decades to come.
Thank you for joining us on this journey into the architecture of your own mind. Until next time, keep diving deep.